The primary purpose of government sterility tests is to prevent the contamination of medical products. According to the World Health Organization, USP sterility guidelines help regulate a wide range of biological medicinal products, specifically in vaccines, blood products, biotechnology products, and cell and tissue products. There are many types of government sterility tests that can detect contamination, but nucleic acid amplification techniques can even screen for viruses. This newer method, combined with previous detection methods, has a high probability of ensuring pharmaceutical products are free from microbial bacteria contamination. Government sterility tests are practical because they can be used in direct contact with medical devices during the incubation period to help test for the presence of microorganisms. Direct transfer is one of the many sterility testing methods that work best for medical devices. Medical devices, solid dose forms, ointments, and creams may be directly immersed in growth media to test for microbial contamination. Membrane filtration is another sterility testing type that assesses the contamination in medical products. Membrane filtration uses a membrane filter unit to retain microorganisms present in the sample article. Then the membranes are incubated in two types of media in order to detect aerobic and anaerobic microorganisms. Both methods require a 14-day incubation period. Sterilization and microbiology testing for medical devices is important because medical devices contaminated with undetected pathogens may become a source of infection for humans. Microbial monitoring is a crucial part of any sterility test. Microbial monitoring ensures that the environment and test samples maintain their immaculacy. Of course, for pharmaceutical products, like vaccines that are used to medically treat viruses and infections, sterility testing is paramount. In the pharmaceutical industry, clean rooms are a highly suggested complement to testing because of their sterile environment. For vaccines, the validation test is needed to determine if the sample will inhibit the growth of microorganisms. The aim of a successful sterility test is stasis, which essentially means that microorganisms cannot grow or replicate within the medical product itself. There are many different types of vaccines, like bacterial, viral, and DNA-based vaccines. Regardless of the manufacturing process or raw materials needed, they must all go through a sterility test to make sure that they are safe for public consumption. To ensure the safety of the public and to abide by good manufacturing practices, these types of sterility testing should be conducted by a certified laboratory, such as Sure-BioChem laboratories. For more information or to receive a quote, visit Sure-BioChem.com. Be sure to follow us and subscribe to our channel for more informative videos.